We know that during acute or chronic viral infection in humans, inflamed tissue, particularly inflamed tissue infected by hepatitis B virus, hepatitis C virus, HIV, liver in this case, or lymphatic tissue in HIV, are infiltrated by several billions of activated cells. So the majority of these cells are not virus specific, but they are activated and undergo apoptosis. And attention, the rate of apoptotic lymphocytes by far exceeds that of apoptotic cells derived from the physiological turnover of tissue cells. At this point, the important question is, which is the fate of this huge amount of apoptotic T cells? Can they become source of self-antigens that are relevant in autoimmunity rather than tolerance, particularly upon phagocytosis and cross-presentation by the dendritic cells? In a series of papers, our paper some years ago, we demonstrated that apoptotic cells derived from activated T cells retain the expression of CD40 ligand, and in virtue of this can stimulate, can induce the dendritic cell maturation that can phagocytose very efficiently dendritic cells. The space that can cleave several fragments derived from cytoskeletal protein that we identified by mass spectrometry analysis, such as vimentin, lamin B1, non-muscle myosin, actin, and these fragments can be exported efficiently into cytosol, processed in, via the conventional class 1 processing pathway, and cross-presented to a huge amount of CD8 positive cells specific to apoptotic epitopes. These CD8 positive cells, in turn, upon performing the effector function, can undergo apoptosis, can be phagocytosis, establishing this vicious cycle that is important in the establishment of chronic immune activation that we demonstrated to be extremely important for inducing AIDS in patients with HIV infection, or to induce chronic infection in patients with HIV infection, but also in autoimmune diseases such as multiple sclerosis or rheumatoid arthritis, suggesting that this mechanism could be a general mechanism of inflammatory diseases. The aim of Ellen Martini in my lab now has been to evaluate if these apoptotic epitope CD8 positive cells are predictive of response to therapy with interferon alpha ribavirin. These cells, these CD8 positive cells, were enumerated in vivo directly by dextramers complex to these apoptotic epitopes, myosin, vimentin, or actin, that were the most immunogenic among several epitopes. As evident, these representative patients showed the higher frequency of these CD8 positive cells as compared to healthy donors. These are both at the level of the single dextramer positive cells or the sum of these dextramer positive cells at the level of single individual. But amazingly, it is extremely important that in longitudinal study during the therapy, we observed a very strong, very significant increase of the frequency of these apoptotic epitope-specific CD8 positive cells after only 1 month of therapy, only in relapsed patients or non-responder patients, but not in other patients such as sustained virological responders or the different types of responders to the therapy, suggesting that the increase of this frequency is related, predict the patient's relapsers or non-responders to therapy. In contrast, dextramer specific for the viral antigens did not display any predictive values, but even they increased after 6 months of therapy only in relapser or non-responders as a consequence of the viral persistence. The antigen specificity of these dextramer positive cells was validated by the finding that they degranulated as detected by CD107 in response to relevant peptide. Altogether, this data suggests that the increase of chronic immune activation due to the expansion of CD8 positive cells specific to apoptotic epitopes predicts relapsers or non-responder patients. But this phenomenon is reminiscent of recent studies about interferon signaling, indicating that in humans a sustained interferon-1 pathway is related to therapy failure. And in these two papers recently published on Science, back-to-back in Science some years ago, that chronic immune demonstrated this paper that chronic immune activation ultimately leads to immune exhaustion suppression and viral persistence. Thus, we, at the right of this data, we suggest that the expansion of CD8 positive cells specific to apoptotic epitopes, chronic immune activation, may be related to the high interferon-1 signature leading to immune exhaustion. Indeed, these CD8 positive cells express very high levels of PD-1, significantly more in relapser than in sustained virological responders. Altogether, this data suggests that a possible involvement of apoptotic epitope-specific CD8 positive cells in disease progression, and that they predict the unfavorable response to therapy in HSV infection. Apoptotic epitope-specific CD8 positive cells can identify profile in patients suitable to new therapy, interferon alpha-3 therapy. And as apoptotic epitope-specific CD8 positive cells express more PD-1 in relapsers and participate to chronic immune activation, these cells may predict patients with a sustained interferon-1 signaling pathway. Apoptotic epitope-specific CD8 positive cells may reveal patients with increased interferon-1 signature, and then could benefit from interferon-free therapy. In other words, these apoptotic epitopes can be used as a biomarker predicting the response to the therapy. Thank you.
There is time for 2 or 3 questions.
Today I haven't heard about autophagy in interferon, but there is a great deal with autophagy in HCV. So, do you have any?
No, I'm not interested to autophagy.
Okay. Then there is another question here.
I think it's impressive how, here in the front, how you get the high CD8 T cell responses against these self-epitopes derived from T cells. And so I was trying to decipher the nomenclature for your peptides, but what are those antigens and why are we not tolerant to them? Why are you getting any T cell response against these self-antigens from other T cells?
Yeah. Only when T cells are activated. Only activated T cells are capable. The repertoire of these epitopes are similar to both activated and non-activated. But only activated T cells can be non-telorogenic, can induce an immune T cell response against these self-epitopes. Because we demonstrated that activated T cells, the apoptotic cell derived from activated T cells, retain the expression of CD40 ligand. And via CD40 ligand they induce this mechanism because they induce dendritic cell maturation. And this has been demonstrated also in vivo.
Yeah, I understand that point of the mechanism, but like you said, the antigens are there already in resting T cells. And these are class 1 peptides that you're looking at. So we should be tolerant to them. And so I'm just wondering why we're not.
Yeah, this is a good point. But many apoptotic cells, I can also only hypothesize why. One possibility is that since apoptosis in the thymus is a large phenomenon, many T cells specific for apoptotic epitopes can migrate in periphery. And however, this is important that the majority of these epitopes derive from long-lived proteins, cytoskeletal proteins, that generally are not processed and presented it's very difficult, unless caspases cleave this protein. This may be a mechanism by which it's possible that this protein can, when cleaved, can become non-tolerant but immunogenic.